Hello, hello. Thank everyone for coming. The topic of my talk will be on the many sources of disease heterogeneity. Okay. So the previous talks really nicely talked about phenotype constructs, analytical choices, sample bias, and these are super important, but they are more methodologically oriented sources of heterogeneity. But I'm going to be talking about how the disease themselves have innate sources of heterogeneity that at some point you just can't get around, right? So I'm a depression predominant researcher, so I'll be using probably too many depression examples in this work. So to explain how the source of heterogeneity at the symptom level can be, I'll use probably the most classic example, which is that to be diagnosed with depression, you have to have 5 out of 9 possible symptoms, which means that there can be 2 patients who only have 1 overlapping symptom, and yet they still have like the same diagnosis, right? So you can imagine that when you are looking at a symptom level, there's this wide amount of variability in what the symptoms they're experiencing are. For the brain, I'm not going to explain to a group of neuroimagers. I think you're probably very aware of the individual variation that the population that you research can have, but I will be talking about this, for the length of the talk. Some sources of heterogeneity that I'm not really going to talk about, but I do just want to mention because I think they're important, is the heterogeneity in genetics. I know that there was a couple of talks, at least, at OHBM that was talking about heterogeneity in genetics, which I think is super cool. There's also, many other sources of heterogeneity across so many different modalities. In inflammation, there's all these different immune markers, all of these things. But, in neuroimaging, at least, when you're trying to find a neural correlate for your disease, at least in depression, there's all of these inconsistent findings, or maybe they're consistent, but they're quite small. I think my favorite example of an inconsistent finding is, this is a depression meta-analysis, but it's a meta-analysis of meta-analyses, a meta-analysis. And they found that even in these meta-analysis results, there is no agreement or overlap in the coordinate-based neural correlates, which I just think is like chef's kiss on the example of inconsistencies. So I don't think anyone's going to be surprised that I'm basically purporting that, at least in some part, that this disease sources of heterogeneity is the reason for these small effects or inconsistent effects. And, I'm not the first one to do this. Several prominent studies have mentioned this. But I did actually show it. Oh, that's so blurry on the big. Okay, anyway, I think it's still readable though. So this is a preprint that I have that I'll have a QR code at the end if you're interested in knowing more about it. But I actually found, so this is a depression population data set, and I have 6 groups that are clinically their clinical profiles are the same, so they're clinically more homogeneous than, say, the circled groups, which is a comparison heterogeneous group where their clinical profiles, their symptoms, are heterogeneous. And you actually can see that pretty much across the board, that red group has smaller effect sizes than the more homogeneous groups. Okay, so if disease heterogeneity is leading to these, small effect sizes, what do we do to parse it? How do we parse this heterogeneity? Mostly we subtype. That's basically the number one thing that we do. This is just literally a couple examples from a bunch of different neurobiological diseases and disorders that do really well done subtyping work. There's truly so much good work in this field, and yet there's really no consensus. So this is, again, some work that I've done where I, in depression, took 4 different subtyping approaches that have been used in previous papers, and in the same subject space, I tried to find how much overlap there was. What is the subject agreement between these clustering approaches? And I actually found that there was almost no. So an ARI of 0 means there's basically no overlap above chance. So I don't think it's surprising then that somehow, even though we have all of this great subtyping work, we have ended up with more questions than answers. So the remainder of my talk will be why is this possibly true, and what are things that we can do about it? So these are the three that I'll talk about. There's many more, but these are just the ones that I'm going to focus on. So for the first one, when we are trying to understand, in this example, brain features as they relate to clinical features, it's maybe not intentional, but just sort of innate in the methods we tend to use. We ask the question, what is the symptom profile to the neurobiological profile, which is a very one-to-one brain-symptom relationship. But it could very well be true that someone could have the same symptom profile, and yet the neurobiological profile that causes that symptom profile can be different. And I also kind of show that. So this is an example of two different clusters where the symptom profile is the same, but the neurobiological profile is different. So there's a red group and a blue group. You can see on the first part of the figure that the red group has increased fractional anisotropy, increased gray matter volume, and increased cortical thickness, not only compared to the blue group, but even compared to controls. So the 0 line represents, that's the black line, represents controls. And then the blue group, the other neurobiological cluster, has worse on all of those, right? And so even though, again, their symptoms are the same, there are important clinical outcome implications to these different neurobiological clusters, because I looked at the cognitive ability of these two different clusters. And again, cognitive ability is not a depression symptom, right? And yet we found that that red group had cognitive ability comparable to controls, but that blue group had significantly lower cognitive ability. So the major takeaway of evaluating this many-to-one mapping is that if you're trying to understand a symptom-to-brain relationship, you have to evaluate both sides. You can't just parse one side and then assume that the other side is then also parsed. Okay, so the second thing that I'm going to talk about is basically the continuous nature of this heterogeneous sample. So again, if we're doing a lot of subtyping, this is usually what we assume to see, which is these well-separated discrete clusters. But actually, it looks kind of more like this, right? So I have some examples of some previous subtyping papers where you can see that these clusters seem to be basically cutting off lines in the continuous variation. And it's not that cutoffs are not, important, especially in a clinical setting. If you have stable, replicable cutoffs, that can be very valuable in a clinical setting. But it also means that we can evaluate the continuation in its own, right? So basically think axes. Instead of having these subgroups, you can think of like principal axes that you can measure to try to understand these relationships, right? So there's multiple different ways you can do that. And people have been doing it and doing a good job at that, but I'm just going to give a couple of examples. So the research domain criteria and HiTOP are some literature-driven ones that come to my mind. This is the HiTOP framework umbrella, and you can see here that they have these continuous signs and symptoms at the bottom, the continuous symptom components and traits. And then they recognize that at the top, there are these higher order dimensions, like these higher order axes. And then, the messy bit is the middle, right? That, they're still trying to figure out is how to relate those two things. But thankfully, we have a lot of data-driven work to try to understand these principal axes. This is just the example that comes to my mind, which is the Xia et al. Paper in 2018 that did a sparse CCA in a trans-diagnostic mental health group and found sort of 4 overall axes that you can relate the dimensionally reduced sort of clinical domain and a dimensionally reduced brain connectivity score, and you're actually able to measure, like represent through this axis that as one increases, how does the other one increase? And then they ended up coming out with these 4. So the major takeaway of this is that not that cutoffs are not valuable, when they're stable and replicable, they are, but we can continue to not be afraid to embrace that continuity. Okay, so the last thing I'm going to talk about is a little bit different than the first two, because the first two are more based off of what seems to actually be going on in the disease and in the relationships between them. But this is a tool that we just need to be doing more and more to be able to understand these sources of heterogeneity. So when I say direct comparisons, I mean that we need to be approaching, like, evaluating these different approaches in the same, like, subject space, for example. So I kind of mentioned before some work that I have done that I actually presented here at OHBM about having the same subject space so that when you are doing these multiple different, parsing the heterogeneity approaches, you are able to have that one-to-one understanding. And then you can have, in the example of my work, I had two subtyping approaches that were driven on symptom data and two subtyping approaches that were driven on neuroimaging data. And I, was able to actually evaluate how much subject agreement there was, but also other things like, what is each of these approaches more sensitive to? What other phenotypes are they sensitive to? And if we really want to be able to understand the relationship between these different sources of heterogeneity, understand the brain-symptom relationship, then we have to actually be comparing the different approaches of parsing the heterogeneity together. And there's not just one way to do this. This is supposed to represent the, you can compare subtyping approaches to a principal axis approach. You can compare literature-driven to data-driven. This doesn't have to be just brains and symptoms. This can also be genetics and all of these other sources of heterogeneity. But overall, the takeaway is that if we want to understand how these sources of heterogeneity relate, we also have to understand and compare the actual parsing approaches as well. So just to summarize everything I've talked about so far, there are many different sources of disease heterogeneity, and there's things that we can do about, and again, these are just a few to sort of get your mind thinking about it, I guess. And if you have any more questions about the stuff that I worked on, I have this, and then thank you, and then the QR codes are still there. Yeah, thank you so much, Janine and Ty, who I have circled here, for inviting me to this talk. Not to out myself being part of the lab, but yeah, thank you so much.